My name is Lynn Cooley. I was a postdoc in Allan Spradling's lab from 1985 to 1988. And after that, I went to Yale University to join the faculty in the genetics department, where I'm now a professor. And most recently, I serve as the Dean of Yale Graduate School of Arts and Sciences, which means I run back and forth between my lab and the graduate school pretty much every day. So I would say the answer to that is partly intentional and partly serendipitous. So as a graduate student, I worked on the transcription and processing of transfer RNAs, tRNAs, and the main project was on a Drosophila gene, but I had never encountered fruit flies. I only worked with genes and in vitro transcription. And as I got further in my graduate work, I came to realize I really did want to work on an animal and have access to genetic approaches and phenotypes and animal development. And right around that time, Allan was invited by the graduate students in my program to come give a seminar in our department. And I knew really nothing about fruit flies at the time, but he gave a very exciting seminar. It was right around the time he and Jerry Rubin had established P-element transformation, and they were doing amazing experiments. So at the potluck dinner that was thrown by graduate students that evening, I got my courage up to go and ask him about doing a postdoc in his lab. And he was extremely gracious, invited me down to interview and meet people and give a seminar. And after a lot of pestering, he decided to let me come to his lab. Well, it was early enough in Allan's lab that he was really at the very early stages of establishing Drosophila oogenesis as the really a powerful model system it has become today. So in retrospect, something that was really exciting about being here was really being at the beginning of establishing fly ovaries as a powerful system. So I felt, now I realize I kind of got in on the ground floor. And I think what I loved the most about being here was that everyone in the lab had their own project, but everyone in the lab was talking constantly to each other about those projects. So you never felt like you were without someone to bounce ideas off of or talk about results, or you just hear what they were doing and that would spark ideas. And that extended both within Allan's lab and beyond, since Carnegie is such an amazingly interactive, collaborative place. So it's like the high point in my scientific kind of growth was being at Carnegie and exposed to that environment where everybody was kind of in everyone else's business in the best possible way. So there aren't that many places that I'm aware of in this country or elsewhere where the principle of being at Carnegie is being in one big group, where reagents are shared. I remember it was stunning to be able to walk to the stock room and just get what you needed and walk out with it. I mean, you didn't have to order it, you didn't have to go get it you didn't have to pay for it in a separate kind of way. You just walked down to the stock room and got it. Well, I would say it's been hard to replicate the idea of having shared open labs. The universities in general tend to be a little more territorial, so labs have their lab space and they tend to stick with that. But I will say that my lab meetings I've tried to make as interactive as they were with Allan, with everybody contributing. We go around the table, we make sure everyone can air their both whatever technical difficulty they're grappling with or discuss their most exciting results and help planning the next experiment. So we have extremely interactive lab meetings. Well, the fruit fly system has always been wonderful. every step of the way that I've been working on fruit flies, but it's hard to overstate how advanced the tools are and how they keep advancing, and how my strong feeling is that the tools that the Drosophila community has developed are at the cutting edge constantly. So being exposed to the broader Drosophila community and being able to do experiments now that we could only dream about when I started working in flies has just been awesome. These days, having access to the advanced ways we can manipulate gene expression, rearrange the genome, and the incredible power of live imaging in just about every project anybody works on it's like being in, I guess it sounds kind of trite or something, but it's like being in a candy store. I mean, you really can just do anything that you can dream of in the fruit fly. And if you can't quite do it, you can either invent a way to do it, or pretty soon someone else is going to invent a way to do it, and they're happy to share it with you.